What additional research would you like to see when it comes to the antibody drug conjugate BCMA-targeted drug that we are talking about? Is there anything additional that we have not covered?
I think for that drug, as we talked about, we know that it has efficacy, so we really need to make it usable. And I'd love to see more research on different dosing options that patients could have, so that the doctors who are prescribing this, including me and you, would have a really good guideline about maybe dose reducing, and if we still have the same efficacy, maybe having things more less frequent dosing, stopping and starting, as you mentioned, and what that means for efficacy. So I'd just like to have a little bit more information and maybe planned clinical trials around different dosing, dose modifications to help us understand if those are still efficacious.
So I agree with that. So how about if you think of like maybe fewer or lower doses or something in combination? What about combination therapies?
Yeah.
Is that something you would like to see?
Yeah, I mean, of course, myeloma is the alphabet soup, malignancy. We put all these letters together to make combinations. And I think one of the great things about the immunotherapies, including the ADCs, but also the bispecifics and the CAR T, is that they lend themselves to combining with drugs that we already have. For example, the IMiDs, because we have a way to stimulate the immune system and work better. There may be even a new role for PD-1 inhibitor, we don't even know. But for the drugs that we do have FDA approved that we could more easily slot in to compare, I think the next step will be, for example, ADC combination with IMiD. I think that would be very outpatient, user-friendly, and there would be a rationale for it.
So given that we sit here talking about ADC, targeting BCMA, we mentioned all these different bispecific antibodies. And I also know you have been leading one of the CAR T cells in parallel in your program. Where do you see the CAR T cells going in competition with all these other antibodies?
I think, yeah, that's a really hard question because I think, first of all, it's going to be a referral pattern based, right? Because not every patient is going to want to come to a center for CAR T. But let's say they had referrals, they could choose whatever they wanted. Let's say it was very free. In that situation, as I see it now, I think maybe the patients would come to CAR T first. And I say that because there's likely to be an FDA approval soon. And then after that, if there was CAR T failure, I think there's still a role for bispecific or BCMA ADC, which at that time they could potentially get in the community. So while they're a little bit younger and a little bit less pretreated in that sense, maybe they can go for something that's more intense as far as logistics and come into a center, but afterwards could maybe also be rescued with a BCMA BiTE or ADC. But I can't predict the future necessarily. That's just how I see it right now.
I think you give a very good overview there. And if I just add one little detail, I think in the end of 2019, the first allogeneic CAR T cells are also coming. So here you have an on-the-shelf product that is still cell-based. So we don't know. This is very early, obviously.
I actually am really excited about the allogeneic product because I think that this idea of off-the-shelf is a big one. When you take people to CAR T therapy, you have to wait, that 5 weeks, and there's a lot of logistics. But if you could just grab some cells off a shelf and give them and they work, that would be great. Of course, there are going to be issues with rejection by the host immune system and issues with persistence. And these are just the things that have to be overcome. But thankfully, the science is moving faster, as you know it already has, than we are in clinical trials. So I'm hoping we'll get an answer to that sooner than later.
And in mice, in our lab, and I'm sure in your lab as well, and other labs, there's work ongoing with the CAR-N case that also can be done on the shelf.
Yep.
So that has not yet been in humans.
Right.
That will be another opportunity for on the shelf for cell-based therapy.
Yes, it will, and those really don't tend to cause graft versus host disease at all. So that's a really good option as well.
So I'd like to thank you, Nina, so much for this very insightful discussion. And I would like to thank our audience for watching this targeted oncology presentation on precision medicine. Thank you very much.